Welcome, everybody, and thank you for joining us in this fairly sunny morning in Seattle. It's my absolute privilege and pleasure to welcome Dr. Poyet and Dr. Iraola, which will open our symposium with the first session on diet, lifestyle, and the global gut. And as Sean mentioned, just wanted to remind you to ask any question that you have in the Q&A session. It's that little button on the bottom of your Zoom screen. And you can also upvote questions if you would like to increase your chance of being asked. So without further ado, let me introduce our first speaker. And Dr. Poyet, feel free to start sharing your slides while I introduce you. Dr. Poyet is working at the University Hospital Schleswig-Holstein, where she is an associate professor in the medical faculty of the Kiel University in Germany. And she is well known for having authored several keystone papers in the microbiome field. In 2016, she co-founded the Global Microbiome Conservancy at the Massachusetts Institute of Technology in the USA, an international nonprofit initiative collecting the biodiversity of human gut microbes to advance microbiome science worldwide. An ecologist by training, Professor Poyet's research focuses on understanding how human-associated microbes evolve and impact human health. So please take it away, Dr. Poyet.
Can you hear me?
Yes, we can.
Good, beautiful. Thank you so much. Great. So first, I have to say that I'm officially still an incoming professor. I was hoping to get my appointment started before this talk, but it's not the case. So let's stay, you know, honest here. And yeah, I'm still in transition from my postdoc at MIT. But thank you so much, Christian, for the introduction. Yeah, it's really a great pleasure to be part of this important session today and to talk to you about some of my ongoing work about the impact of industrialization of human lifestyles on human gut microbiome. So before that, I actually would like to make an opening remark about what we know about the human microbiome. The next generation sequencing technologies developed over the last few decades really contributed to advance our knowledge. And this is exactly the topic of our session today, even if we clearly start to know a lot, particularly about how macrobiota affects host physiology and how perturbation in the gut microbiome associate with many non-communicable disease, for example, including inflammatory bowel disease, cancers, cardiovascular disease. The vast majority of our existing knowledge is still derived from studies focusing on the microbiome of industrialized population, mostly Caucasian people living in the US and in Europe. And this is actually very well illustrated here on this figure published earlier this year by Richard Abdil from Ram Blackman groups. Where you can see the important imbalance that exists in microbiome science. The number of microbiome samples per country and the cumulative microbiome sample per world regions, with most of the sample coming from Europe and North America, and also the over and under representation of each country worldwide, and the annual proportion of sample per region, with again a very important underrepresentation of the less industrialized low and middle income countries in microbiome science. But actually, we already know that the very well represented industrialized gut microbiomes associate with most of the non-communicable disease existing worldwide. As you can see here, when looking at the spread of non-communicable disease occurring more frequently in industrialized areas of the world. So not only the microbiome science is highly biased, but the drawing of the human gut microbiome that we are making is also based on potentially disturbed microbiomes that associate with specific and industrialized lifestyles and non-communicable disease. And to end this very short introduction, I would also like to point out that the United Nations recently predicted that 70% of the human population could live in urban areas by 2050. which means that many human communities could soon change their lifestyles and adopt some degrees of industrialization, which could modify their microbial diversity, and with it generate the rise of microbiome perturbation and potentially non-communicable disease among this population. So basically, if we want to better picture and understand the human gut microbiome and its impact on human health, and if we want to counteract the unethical imbalance that exists currently in microbiome science, and that could lead later on healthcare inequities, basically because the underrepresented groups will be less likely to benefit from scientific advances tailored to the most studied population. Well, if we need to, if we want to counteract for all this bias, we need to promote the diversity of human population included in microbiome science, and we need to do it pretty quickly. So for all these reasons, back in 2016, in the laboratory of Eric Alm at MIT, Mathieu Groussin and myself created the Global Microbiome Conservancy, so GMBC. And so this initiative is a non-profit collaboration between more than 80 scientists from 30 countries around the world with very complementary backgrounds, with microbiologists, anthropologists, clinicians, and we are all working together and biobank and make available for research the biodiversity of human gut microbiomes to advance microbiome science worldwide. Equitably and ethically. So together, we already recruited 1,300 participants across a bit more than 40 human communities worldwide. So the yellow dots show the location of all the population we already worked with, and the blue dots show the country where we're starting new collaboration with local scientists and plan to recruit participants in the next few years. So we recruited individuals among both industrialized and non-industrialized countries, as you saw. And in each country, we also recruited individuals among both urban and rural groups. We recruited only adults who do not have any obvious signs of infections or known disease. So the distribution of demographic parameters are shown on the bottom left and are typical of relatively healthy human population. And here I'm also showing you the genetic diversity of our cohort so far, which we characterize by exome sequencing and genotyping, and which we compare to the genetic diversity of the 1,000 Genomes Project. And as you can see, our cohort represents a wide range of populations that have genetically that have been genetically defined so far. So we gathered stool samples from each participant, and we generated multi-omics sequencing data that allowed us to look at really the impact of lifestyles, and particularly the level of industrialization on human gut microbiome. And so on this plot, where basically individuals are colored by subsistence strategies, so the blue dots being the most rural and isolated non-industrialized individuals, and the red dots the more urban and industrialized individuals. And you can see how really lifestyle strongly associate with specific gut bacterial communities, which are drastically different in the gut of people living rural lifestyles in blue, or people living more urban lifestyles in red. So on this plot, based on metagenomics taxonomic profile. And as you can see now, not only the composition is different, but the diversity of bacterial species living in your gut is also significantly reduced with industrialization. And I also wanted to show you these results because the microbiome is not only the bacteria, but many other things. And so I wanted to show these results generated in collaboration with Malte Hohlmann at the KMB at Kiel University in Germany, showing that the same trend is also observed when looking at the virome diversity and composition. The virome also highly associates with host lifestyles. So here you have the same color code, and the diversity is also reduced with the more in the most urban and industrialized populations. And finally, so using the stool samples processed in the lab, we created a pipeline to generate high quality biobanks of pure bacterial isolates and their associated whole genomes. So using anaerobic chambers and by designing a wide range of culturing conditions, we built the GMBC biobank, which is the most diverse biobank of human gut-associated bacterial isolates available for research community. And as you can see on this tree, our biobank regroups hundreds of species, including many unknown and many previously considered unculturable bacterial species. And finally, last month, the Global Microbiome Conservancy started a collaboration with another nonprofit organization called OpenBiome and located in Boston in the US to build up the initiative. So the GMBC program at OpenBiome will now be led by Katia Moniz to continue and expand the collection of samples, the biobanking of bacterial strains, and the distribution of biospecimens to the research community. So I highly encourage you to check our website to know more about our work. And if you are interested in joining our effort, please feel free to contact us. But so now I would like to give you two examples of stories that we can go after with these amazing global data sets and biobanks. So the first one about the impact of industrialization on one specific example of host microbiome interaction. So it's a collaborative work with Mathieu Groussin, so the co-founder of the GMBC and new associated prof at Kiel University as well, and Tu Nguyen with a postdoc in American lab at MIT. And so for this first story, we looked at secretory IgA measured from stool samples. So very briefly, intestinal IgAs are tightly connected to the microbiomes. Once secreted into the lumen, they promote the exclusion of pathogens and antigens. They also interact with commensal cells to prevent overgrowth of biofilm formation, for example. And they also promote the colonization of beneficial bacteria within the mucus layer. So we were curious to know whether the level of IgA secreted, which are directly influenced by the intestinal microbiome, change across population with different gut microbiomes. And actually, when we looked at the level of IgA in the stool sample that we collected from our worldwide populations, we found that the IgA level actually strongly associate with differences in microbiome composition between people, explaining about 7% of the variance, which is illustrated here in this multivariate plot showing differences in microbiome composition between people with individuals colored by a level of IgA. And so the more pink you are, the more IgA you have in your stool sample. And actually considering that usually individual host factor correlate to the microbiome in population-based studies explain about 0.1 or 1% of the variance, this is actually a very strong association. And actually, very interestingly, we also found that this association between IgA and the microbiome composition actually recapitulates the association with host lifestyles. As you can see here, industrialized individuals secreting much higher level of IgA in their gut as compared to non-industrialized individual with a difference of about 1 log. We also observe this difference at a higher resolution, as shown here on the right. So here we compare the IgA level between urban in blue and rural individuals in yellow within each country. And so even at this resolution, we can see significant differences in IgA levels with urban individuals secreting higher level of IgA in their gut. And actually, if you think about it, this trend is counterintuitive, as non-industrialized individuals are actually more frequently colonized by eukaryotic pathogens and parasites, which we actually confirm with our data. So many of these parasites are actually targeted by IgA, but the difference we are seeing here goes the other way around, and maybe related to differences in antigenic nature of commensal bacteria in the microbiome, maybe. So next, we were curious to know if the bacterial species actually coated by IgA in these samples could be different between individuals of different lifestyles. So to check for this, we used a technique called IgA sequencing, which allows for the identification of bacterial taxa through 16S sequencing of the specific cells, bacterial cells that are coated by IgA. And so here, because we had hundreds of samples to check, we use a relatively high throughput magnetic beads-based assays developed by Noah Palm to sort IgA coated cells. And so we calculated an IgA coating index for each bacteria, representing basically the abundance of the bacterial species in the IgA positive fraction over its abundance in the IgA negative fraction. And so we consider taxa with an index higher than 10 to be quoted, or let's say strongly quoted by IgA. And so with this data, we found that the proportion of IgA-coated bacteria is actually higher in the industrialized cohort compared to the non-industrialized population. And this difference being in the same range of as the one that we observe between healthy or disease individuals in multiple diseases, actually, such as IBD, multiple sclerosis, for example. And we also found that the intensity of the coating profile is stronger in the industrialized microbiome, as suggested on this plot on the right. So in conclusion, there is a higher secretion of IgA among industrialized population, with more bacterial species being targeted by IgA and with a stronger intensity. This really suggests that changes in industrialized microbiome composition also impact host microbiome interactions and our immune system. So we now need to mechanistically understand what's going on here, what's driving this increase of IgA. For example, it could be the results from a higher level of pro-inflammatory signal, or result from higher exposure to gut bacteria in general due to increased impairment of the gut epithelial barrier, for example. So we are currently trying to answer this question with additional experiments, including in animal model. For the second story, I wanted to talk to you about the impact of industrialization on one very specific gut microbiome function, because whether changes in microbiome composition between human population results in differences in microbiome functions is still largely unknown, despite the fact that this is really what we need to know to understand potential impact on human health. So that's a work that I'm leading in collaboration with Mathieu again, but also Enara Sisiaga from the University of Copenhagen and former postdoc in Roger Simmons' lab at MIT, and Antonio Valencia, postdoc with Ramnik Xavier and Emily Balske's lab from the Broad Institute and Harvard. So this specific function that I will talk about is the degradation of cholesterol driven by the gut microbiota and its potential effect on chronic disease, such as cardiovascular disease, for example. So very briefly, the cholesterol circulating in your intestines comes from, for a minor part from your diet, but also directly internally from your liver, mostly in the form of bile acids. And then about half of this cholesterol is reabsorbed by the intestinal epithelial cells back to the liver, and about half is excreted in stool. But what is less known is that the cholesterol can also enter a conversion pathway thanks to specific gut bacteria. And this pathway converts cholesterol to another sterol called coprostanol, which is only produced by bacterial metabolism. And contrary to cholesterol, coprostanol is not reabsorbed through the lymphatic and blood system, but is entirely excreted in stool. So in theory, if the conversion of cholesterol to coprostanol in the gut microbiota is efficient, then the quantity of cholesterol being reabsorbed back to the liver will be reduced, and a higher level of coprostanol will be excreted in stool. So again, in theory, engineering the gut microbiota so it can convert cholesterol to coprostanol in the intestine and so reduce cholesterol level in blood is of great potential in the context of high cholesterol related disease. And if this works, one easy way to measure the efficiency of cholesterol degradation will be to measure the ratio of coprostanol over cholesterol in stool, because this ratio will be higher when the conversion is basically more efficient. So one of the first things that we did using the Global Microbiome Conservancy biobank of stool samples was to compare this ratio of coprostanol over cholesterol in stool samples. And to make it simpler for this talk, I categorized all the population that we have in just two categories here, rural non-industrialized and urban industrialized populations. And to our surprise, by measuring this ratio of coprostanol over cholesterol in stool sample, we found that non-industrialized individuals have much higher coprostanol over cholesterol ratio than industrialized individuals, suggesting a more efficient conversion being performed by their microbiota. But because these preliminary results could have been driven by many different factors, we then conducted an ex vivo experiment using the same batch of stool samples. So concretely, we put small amount of stool in a tube containing a bacterial media enriched in cholesterol. And we measured the coprostanol over cholesterol ratio in each tube over time. And as you can see here on the right, the result we obtain here, for example, after 8 days, confirmed that the microorganisms present in the stool samples of these non-industrialized individuals are more efficient at turning cholesterol to coprostanol. So both these first results suggested that gut microbiomes of non-industrialized individuals are better at converting cholesterol, and that this function may have been lost actually during transition to industrialized lifestyle. So naturally, we then wanted to identify the very species capable to carry out this metabolism. But historically, cultivating coprostanol producing bacteria have been very challenging. As of today, only one single bacterial strain named Bacterium coprostanoligenes and isolated many years ago from pig sewage is available for research. And only one gene of this conversion pathway encoding for a cholesterol dehydrogenase and named ISME has been characterized using these non-human associated bacterial strains. So using the sequence of this characterized ISME gene, we run a genomic screen using the global Microbiome conservancy bacterial isolate collection of whole genomes. And we were very excited to identify for the very first human-associated bacteria and bacterial cholesterol converter candidate in our biobank. So this isolate is a firmicutes, genomically very close to the E. coprostanoligenous strains and possess a highly conserved dehydrogenase gene in its genome. Using the metagenomics data that we generated from each stool sample, we analyzed and we confirmed that actually the stool proportional over cholesterol ratio is positively correlated to the abundance of our candidate cholesterol converter. That's a good news. And we also were able to highlight that our cholesterol converting candidate is less and less abundant in a population that have higher degrees of industrialization and urbanization in their lifestyle. So as you guess, we next tested this specific candidate strain in vitro. So it was about the same setup as the ex vivo experiment, but this time we used individual isolated bacterial strains in presence of our cholesterol-enriched media. And so actually, we were extremely excited to confirm the efficiency of our candidate bacteria to turn cholesterol to coprostanol in vitro, with the ratio of cholesterol decreasing over time while the coprostanol increased. In parallel, we tested bacterial species from 32 different genera, which don't possess the ISME gene, and none of them actually show any sign of cholesterol conversion. But then to look for additional candidate cholesterol converters in the lab, we used our ex vivo approach using complete microbiome communities as described earlier. And so stool samples were incubated with cholesterol, and we integrated longitudinal metagenomics data with metabolomic data to detect taxa associated with coprostanol formation. And you can see here on the right the list of these detected taxa that associate with the coprostanol formation. And so first, we were able to recover our previously characterized coprostanol former, so E-copro in purple, so that's good news. But we also found a group of species shown here in orange that are all part of the Ocylispiraceae family. So this is pretty exciting because it's one of the unique group of bacteria that has been found to be associated with obesity in meta-analysis of microbiome sequencing data. And so we then looked at those species specifically. And so we grew this Ocylispiraceae species alone with our cholesterol-enriched media to check whether they were efficient at producing coprostanol. And actually, when comparing the absolute concentration value to those obtained previously with our E-copro candidate strain, we measure only, as you can see, only a very little amount of coprostanol. But however, when we grow both taxa together, so E-copro and Ocyli together in co-culture within the same tube with the cholesterol-enriched media, we now see a huge depletion of total sterols in the media. So it looks like the production of coprostanol is not that good, but the total sterol, including cholesterol, level drop drastically, about 2 log in concentration, and is actually almost completely removed. Okay, so yeah, we have ongoing experiments, obviously, including the characterization of this co-culturing effect to optimize the cholesterol conversion, RNA-seq generation to characterize the other genes involved in the conversion pathway, and also very promising in vivo experiment that actually seem to highlight the reduction of both fecal but also serum cholesterol in mice fed with our bacterial strains. So yeah, I'm going to end with that, but I think altogether these results strongly suggest that our lifestyle not only influence the composition of our gut microbiome with specific bacterial species missing from the gut microbiome of industrialized population, but also has impact on the presence or not of some function that can be particularly relevant for human health, like this one related to the degradation of cholesterol. Okay, so I would like to thank Mathieu Groussin, who co-lead with me all the work that I presented today, as well as Eric Alm, all the AMLab members, all the GMBC consortium members and participants. And I would like to thank our collaborators that help with the cholesterol proportional GC and LCMS measurements from Rami Xavier Emily Balskus lab, particularly Doug Kenny and Antonio Valencia, and people from Roger Simmons' lab, and particularly NRS again, Fatima Hussein. Yeah, and I thank you all for your attention.